Okay, so I'm going to start to introduce you, Doris. So it's my pleasure to introduce Doris today for this monthly webinar of EUROTOX. First, I want to thank you very much, Doris, for accepting to give this presentation. So the presentation is, the title is Food Toxicology and Mycotoxins. And Doris, she's a professor of food chemistry at University of Vienna in Austria and head of the Department of Food Chemistry and Toxicology. She studied food chemistry at the University of Kaiserslautern in Germany, and she's a member of several national and international committees in the field of food safety. In her research strategy, she synergistically combines state-of-the-art analytics with toxicological mechanistical approaches to address actual questions on consumer safety. Actual fields of research include hazard characterization of emerging contaminants, the complex interaction of contaminants which potentially co-occurring bioactive food constituents of pharmaceutical drugs, as well as novel strategies for risk assessment of chemical mixtures. I think this presentation is really timely because there's a lot of question and concern about contaminants in food. So again, thank you very much, Doris, for accepting to give this presentation. So the floor is yours. And for the participant, please put your question in the chat, and we will have 15 minutes at the end of the presentation for the question. Thank you. And plus, and turn down your speaker, please.
Yeah, thank you, Mark, for the very nice introduction. And it's my pleasure to talk today about one of my most favorite fields of research, mycotoxins. And I tried to cover state of the art and give you a little bit of a glimpse what's going on in the scene and what are the challenges we are facing in near future. So the question is it an old thing? Is it a new thing? Well, mycotoxins are around for quite a time. So their first mentioning of mycotoxin effects and intoxication even in the first part of the Bible. And you may all be aware of these very famous paintings. These guys, they have lost their feet and legs, and these people are severely suffering, and they are suffering from toxicosis, from intoxication with ergot alkaloids arising from consumption, presumably of molded ray. And these intoxications with ergot alkaloids, though the main intoxications were observed between the medieval times or even earlier until the 19th century. Nowadays, with modern methods, it's kind of rare to see a real strong intoxication. But still, there are these infiltrations of our ray. That's a picture of one of my colleagues from grains from near Vienna, and you see these black corns. These are Claviceps purpurea corns, which are heavily contaminated with respective ergot alkaloids. We have to sort them out, and we are on a good way here. So, well, the real research in mycotoxin research started more or less in 1960, when in the UK, a tremendous effect was seen on turkey farms, and more than 100 or about 100,000 turkeys died due to liver failure. And in the end, it was found that these fatal effects came from spoiled peanut cakes used for feeding these animals. And they were spoiled with Aspergillus flavus. And then it was possible to identify the main constituents, and this is the most famous mycotoxin by now. This is aflatoxin B1, and we will come to that a little bit later. And so, well, you could say, oh, 1960, aflatoxin B1 was then found, and we are now nearly more than 60 years later. But despite improvement of good agricultural and manufacturing practices, we still do have mycotoxins in food, and sometimes in very substantial amounts. But here we have to see, where is the infection coming from? Is it on the field? Is it during or after the harvest, or is it in the storage? And this is of tremendous impact on the compounds we can find. And that point I will make a little bit later. But the main mycotoxin producers we know by now are Aspergillus, Fusarium, and Penicillium species. So, well, you have to see, since 1960, in 1960, we were so happy if we could identify one compound. In the 1980s, oh, if we could identify in parallel 6 or 7 compounds, oh, we were fantastic. So nowadays we have multi-methods for over 1,500 compounds at the same time. And well, with that, we have new ideas about what is really in our food. And we can say that nowadays we have estimation that up to 90% of the global food crops are at least contaminated with some trace levels of mycotoxins. And sometimes it's not only traces. Yeah. And we have another contributor, and we should absolutely not underestimate that. And this is the climate change. If the climate changes, the conditions for the growth of the mycotoxin formers also changes. And so the pattern of contamination will change. So how is the regulation at the moment in Europe? What is regulated and what is monitored? From the around 1,500 compounds you can analyze, only more than a handful are regulated and monitored. And of course, here you see the aflatoxins, but also several others, ochratoxin A, patulin. And then you have a whole bunch of compounds, and they are generated by so-called Fusarium species. And this is an example here in the picture of Fusarium graminearum. So you can find them on wheat, on corn, for example, and these are the most predominantly targeted grains. So coming back to the aflatoxins, let's have a closer look to the different toxins. And I would like to go from the very well-known ones to the ones which are popping up and demanding more research. So what do we know about aflatoxins? So we know since quite a long time that aflatoxins are potent liver carcinogens. And this is as old as you can see, this is a monograph from IARC from 1976, associating the intake of aflatoxin with the rate of liver carcinomas per 100,000 inhabitants. And you nicely see there's a very nice correlation. And we now know, meanwhile, that this is indeed a causal connection. So, well, in that graph, you saw all these African countries. And so what may be contributors? Of course, something like more traditional food, cassava, for example, but also food which is nowadays found in our food chain, also in Western countries. Yeah, so peanuts, peanut butter, peanut oil, and you can generate a lot of things out of peanuts. Then, of course, corn, maize, sweet potatoes, and these are substantial contributors. So is that always plant-based food which might only be affected with aflatoxins? No. So the point is that if feed is contaminated, and for example, the cow would ingest respective contaminated food, then a modified form of the aflatoxin, the metabolite, for example, M1, is excreted in the milk and may get into the food chain. But of course, M1 can also be found if the breastfeeding mother eats contaminated food. So M1 might occur in respective breast milk. So what is aflatoxin then doing? So, and the most important structural element in that respect, this is aflatoxin B1 and the double bond in 8-9 position, that's the crucial one. So in the liver, upon metabolization in phase 1, an epoxide is formed, and that very epoxide may attack now the DNA, may form DNA adducts here at predominantly at N7 of guanine. And if that ring is opening, here you see the opened ring, then you generate a very, very stable, very mutagenic adduct, the so-called FAPI adduct for formamidopyrimidine adduct. And that epoxidation in the liver is due to, of course, cytochrome P450 enzymes, which can be 1A2 or 3A4. Yeah, and we generate very characteristic pattern of respective mutations, and one of these hotspots we know since quite a long time is a hotspot mutation in the P53 gene in codon 249. Yeah, we have regions in the world which have quite a high rate of aflatoxin of liver cancer, together with quite a high exposure with respective aflatoxins in food. And that marker or respective markers can associate whether aflatoxins contribute here. So taking that together in a nutshell, so aflatoxins, especially the ones with a double bond, like aflatoxin B1, act as liver carcinogens. But beyond that, there's quite accumulating publications indicating that this phenomenon of stunting you observe in certain regions of, for example, Africa, might be associated with exposure in utero to aflatoxins and early life exposure. And we also know that hepatitis B virus infection may also contribute, together with exposure to aflatoxins, to the onset and development of liver cancer. So what are, for Western countries, potential contributors to exposure? Of course, all products containing maize, but also groundnuts. Pistachios might be a big bringer, almonds, figs. You know, figs are harvested usually very ripe, and what is ripe can also be a little bit pun ripe and may then have an open door for respective spoiling fungi, but also other cereals might contribute. So we say that aflatoxin forming species, respective Aspergillus, need high temperature and moisture. Is Europe really at risk? Yeah, beyond import? Yes, indeed. Climate is changing, and there have been already publications on aflatoxins in wheat in the south of France, for example. So we have to be very aware of the respective impact of climate change. So that was aflatoxins in a nutshell. I would say this will always be something which will accompany us, and we have to control it. What we also have to control is another mycotoxin, EUROTOX A, which is also a carcinogen. It is nephrotoxic, not so much liver, but nephrotoxic. And this is now a big spectrum of food which might bear EUROTOX A. Several cereals, but also all corn, rice, but grapes, so might find the way into wine. Then dried fruits like raisins, but also coffee beans, cocoa beans, and that means coffee, chocolate, tea. So, and EUROTOX A may be formed also under moderate climate conditions. And also, like aflatoxins, there may be a carryover from contaminated silage feed. But in a different way, in here, we have to consider that ochratoxin A has something very special. It has a very unique structure. You have a chlorine here. You have phenylalanine as a part of the molecule. And ochratoxin A has a very, very unusual half-life in the bloodstream. It has a very, very high affinity to albumin, and that means it can stay in the blood of humans or animals for days. So we have a very, very slow elimination time, and that means if you consider material like in Great Britain, black pudding, or the famous Blutwurst in Germany, or respective products where you use a lot of blood, then if that's contaminated, then ochratoxin A may also find the way into respective products. Interestingly, the whole mechanisms of action of ochratoxin A have not been fully elucidated yet. We know it's nephrotoxic, but it has also some immunosuppressive and teratogenic effects, but it doesn't appear to generate DNA adducts. Yeah, so it's quite a complex mechanism, which is still under elucidation in many groups worldwide. So that were the carcinogenic ones, predominantly carcinogenic ones, liver cancer, kidney cancer. So let's have a look into the fusarium toxins. So fusarium organisms attack already on the field, like you can see that here with a famous fusarium wheat blight. Yeah, so we have different types of toxins. You see the structure is very, very different from what you have seen before. They do not generate DNA adducts. That's very different. They attack predominantly cereals, so wheat, maize, barley, oats. So, and one type of fusarium toxins are the so-called trachlotethins. Trachlotethins have 4 different types, and the A type consists of so-called T2 and HT2 toxins. And these toxins have been found responsible for very severe intoxications, so-called alimentary toxic allergia, which was observed in the former UdSSR during World War II and thereafter, when people were starving and had the pressure to eat cereals. Also, they were maybe moldy because they were overwintered. So the symptoms are vomiting, abdominal pain, diarrhea, followed by leukopenia, and the lethality is very, very high. So T2H2 is, well, an old story, but it's also an actual story. For example, at the end, I think of January, we had this alarming information in Austria that T2 and HT2 was found in several oat cereals for breakfast. So 4 out of 13 packages from the supermarkets in Austria were found to contain T2 and HT2. It strongly depends on the weather conditions in the year, but that really has to be controlled. T2H2 can't be eliminated at the moment, and especially in oats. That's a big point. So if we look into the grains, wheat, maize, barley, oats, and rye, these are really important cereals for our daily food. So, and a little bit a different structure is the B-type deoxynivalenol, also belonging to the fusarium toxins. And it's also called vomitoxin, and that nicely describes acute intoxications. So this is one of the most abundant mycotoxins in temperate climate regions. in cereals. And this is they are found in quite a high amount in these so-called Fusarium head blight wheat samples. So what is the mechanism of action? And this is quite interesting. It's, again, not DNA damaging per se. No. Deoxynivalenol inhibits the generation of proteins at the ribosome. It's an inhibitor of the peptidyl transferase, and with that, it's very cytotoxic because it doesn't allow anymore to have a synthesis of the high throughput proteins in the cell. So, and if you look into the structure for a moment, then, well, this is kind of water soluble. That means it acts to quite a substantial amount at the direct target tissue of the intestine. And there it might be very cytotoxic. And it's also known to be very pro-inflammatory. So, it's not very good for the intestinal tissue. And of course, it can also damage after uptake the liver. But the local effect in the intestine can be quite substantial. Yeah, well, and one of the last types of mycotoxins identified and now already regulated, generated by Fusarium, are fumonisins. They are, again, completely different in structure. It's a little bit resembling to fatty acids, are these long chain things with acids here. So they are hepatotoxic and nephrotoxic, but they don't form DNA adducts. It's speculated they might contribute to esophageal cancer onset, but that's not really clear. But it may affect the uptake of folate, and with that, it contributes to the development of neural tube defects in the developing fetus. The predominant mechanism of action of fumonisins is now again completely different to the compounds you have heard before today. Fumonisin B1 has been shown to disrupt the sphingolipid metabolism. So it is an inhibitor of ceramide synthase, and with that, dramatically affects our sphingolipid metabolism. So completely different from all other mycotoxins you've heard before. So, and now we can even make the spectrum of mechanisms a bit broader, and we go to the last class of Fusarium toxins for today, the zearalenone and zearalenone derivatives. Completely different again, no DNA adduct, in that case, no sphingolipid effect, no, but it is an estrogen. Zearalenone is formed by Fusarium species and has estrogenic properties. And if zearalenone even is reduced, that the keto group here is converted to a hydroxy group, generating alpha-zearalenone, this enhances estrogenicity by a factor of approximately 60. So, and we know very well, for example, in pigs, they are very susceptible to these estrogenic effects that respective spoiled feed is associated with reproductive problems. So we have here a compound which needs to be controlled because it acts as an endocrine disruptor. So that was a little bit of a spectrum of important regulated mycotoxins. So the question is, for toxicologists, what is really reaching the human organism? So the fungus is generating a toxin, and we are at the end of the line. So, but if the fungus is attacking the respective food they want to live from on the field, and the plant is still living, then the plant may also have a metabolism and may change the composition of the toxin. And of course, food processing might also change the toxins. But in addition, well, make it more complicated, the fungus is generating a toxin, the plant might metabolize it, an animal might eat that, and the animal might change it, and then food processing is coming into the picture. Well, we cannot, unfortunately, talk to all about all the points here, but I will give you a little bit of a glimpse on that plant metabolism thing, because as a toxicologist, often focusing on mammalian systems, we do not always consider plants to really contribute. But a plant has also respective metabolism, but it's a little bit different from what we are able to do. So what do we do with a compound we want to get rid of? Well, if phase 1 metabolism is not necessary, then we conjugate and we excrete, yeah, either with feces or with urine, or with sweat. Well, consider a plant cell. A plant cell which is attacked, a toxin is formed, enters the cell. Well, that poor plant cannot set away urine. So, but what can the plant do? So, for example, with deoxynivalenol, also a plant has ribosomes. It's fatal for the plant cell if such a ribosomal inhibitor is really present in the cell. So it has to be detoxified. So the plant is able to either add a sulfate or a sugar. Especially glucose is very often used. So the plant can conjugate, but not excrete. So, but the plant has a vacuole. So the conjugate is then transported to the vacuole and stored there, detoxified there for the plant cell. But if we eat then the corn, or the animal eats the corn, what do we eat? We eat the remaining amounts of non-transformed toxin, and we eat the conjugated ones. And if there's a sugar conjugated to such a compound, then it has a different kinetic in our gastrointestinal tract. So it has been demonstrated for several of the fusarium toxins. Fusarium attacks on the field. The plant is still living. So the mycotoxins like deoxynivalenol or thearalenone can be conjugated to glucose with respective glucosyl transferases in planta. And then we have quite a water-soluble compound. And that one is not absorbed in the small intestine of humans or animals as the free toxin. So it might then reach the colon, where microorganisms might cleave away the sugar, and then in the colon, absorption might take place or not. Kinetic is changed. So this is a small glimpse, and it's a big issue because many modifications might take place already in the plant. And then we have all the animal metabolism. If the animal is eating respectively these kind of toxins with the feed, and then metabolism may occur in the animal, and we have a very complex analytical duty. So this was, in a nutshell, the regulated mycotoxins more or less. And I hope I could demonstrate that we have so different topics. So we have the aflatoxin generating DNA adducts doing stunting and being really, really liver toxic. EUROTOXin A, which is nephrotoxic, generating kidney cancer. I let out patulin because it's not so extremely dangerous, but it's especially of interest because the biggest source for patulin is apple and apple juice and apple cider. And so we have to keep that under control. But then we have this big part here of fusarium toxins, ranging from inhibition of protein synthesis, estrogenicity for zearalenone, and sphingolipid metabolism inhibition, and the ergot alkaloids I mentioned in the beginning. But, well, I said we can analyze much more substances nowadays, and there are so many not yet regulated and monitored secondary metabolites generated by fungi. And from them, the most, at the moment, one of the most famous ones are eniatins, alternaria toxins, and bovaricin. What are emerging mycotoxins? These are compounds where data on occurrence and toxicity are not sufficient to allow the comprehensive risk assessment yet. So, well, they are heavily investigated, and so eniatins are very different in from the structures you have seen until now in my presentation. They are these cyclic peptides, and they differ then in these residues, and they have a very interesting spectrum of toxic activities. Also, no indication for DNA adduct formation, but very complex profile with respect to activity. So I have to focus today a little bit, and I will tell you a little bit more about a field we are heavily working on since quite a time. And this is another class of important emerging mycotoxins, the so-called alternaria toxins. Alternaria are very special guys. And one of the important strain is called Alternaria alternata. And they grow under a big spectrum of conditions. So from very nice warm 25 to 28 degrees and being quite humid until the conditions in your refrigerator at home. So if you have not very, I hope not, but sometimes refrigerators are not always very nicely maintained. If you have these black spots in your fridge, it might occur that it's Alternaria alternata, and it might generate toxins. So, and as you can see, Alternaria alternata can grow on things which are stored, but it can also grow on living organisms, like that overwintered wheat, for example, tomatoes, carrots, a whole huge spectrum of food items. And of course, if you have the choice, you wouldn't eat that tomato and you wouldn't eat these carrots. But what do you do with dark spots on dark fruits? That's kind of tricky. So there was a publication some time ago about quite a high rate of Alternaria alternata found on ready-to-eat berry fruits in American supermarkets. So dark spots on dark fruits are hard to spot. Of course, you don't eat this tomato and that carrot, but how about that tomato sauce? You don't see how that tomato looked like when it was prepared for. And this is one of the big problems we have identified, that some of these preparations, tomato sauce, canned tomatoes, tomato paste, and so on, that some of these products contain quite interesting amounts of these toxins. But which toxins are we talking about? And now things get really complicated. We have a whole spectrum of different toxins, from very famous ones like altanariol here, dibenzopyrens, and here the respective, they just differ in that methyl group here, the monomethyl ether. Then we have tenoarsonic acid and tantoxin, very different again. And very interesting class, very complex one, the so-called perilane quinones, alta-toxins, and stem cell toxins. So unfortunately, only a few of these compounds are commercially available. And what you can see here is that also glucosides and sulfates can be formed, especially when active living fruits are attacked. That may be an intact tomato on the field or in your fridge, or it can be also sunflower seeds. It may also be wheat or other commodities. So what do we really find from these toxins in food? So we did some analysis some time ago, and my colleague Benedict Ward set up a multi-method for that together with his PhD student Hannes Puncher, and analyzed tomato products, sunflower seed oil, and flour from different countries in Europe. And to sum up a little bit, what you see on the first glimpse, this is tomato sauce, sunflower seed oil, wheat flour. And you quickly see it's never a sample with only one toxin. So it's always a mixed contamination. Tenoarsonic acid is always the highest one in all three categories. Well, in the sunflower seed oil, you do not see the sulfated compounds. Of course, they are more water soluble, but they are found to substantial amounts in tomato sauce. Yeah. And from the perilane quinones, we found in all three categories samples which are contaminated. So respective data have been published in food control to give an idea about this mixed contamination. So taking all the studies we have done in that context together, I can say that tenoarsonic acid, altanariol, and its monomethyl ether, they are indicators of infestation with Alternaria, but they do not allow any conclusion on the content of other Alternaria toxins, especially on the content of perilane quinones. And if we go quickly back to the structure, then you see we have structures here with natural epoxides. And these natural epoxides are extremely genotoxic. They form DNA adducts. And the question is, do they reach our food chain or do they react before? Yeah, that's a big, big question. Okay, so the mechanism of action. What are important effects of Alternaria toxins? With respect to genotoxicity, I already mentioned AOH and AME. They are kind of weak. They don't form DNA adducts, but they interfere with topoisomerases and lead to DNA strand breaks, but that's not very potent. But these alta-toxins, alta-toxin 2 and stem cell toxin 3, they are very, very potent due to these epoxide residues. They form DNA adducts. So, but we cannot exclude that other compounds in that big mixture they generate also contribute to genotoxicity. So some of these, we found one food item so far with really a high level of alta-toxin 2, and that was that black spot on that very apple. That was kind of interesting because here we had very, very high concentrations of alpha-toxin 2 in that apple. So don't eat your black spots on the apple. So far for genotoxicity. So the second topic for alternaria toxins is endocrine disruption. And why so? We know that alternariol and also its monomethyl ether are moderate mycoestrogens. They have estrogenic activity. Interestingly, a whole big mixture of a culture of alternaria toxins do just the opposite and generate antiestrogenic effects. And we are currently investigating the rationale behind and try to find the compound which is responsible for that effect. So we have endocrine disruptive effects, which is not fully elucidated to allow a full risk assessment. And the last topic is, from my point of view, a very important topic. It's the topic of immunotoxicity. So some time ago, we could demonstrate that macrophages, which are derived from THP1 cells, and you stimulate the immune response with LPS, then when you add subsequently alternariol, then you suppress the immune response quite effectively. So we investigated whether that's also an issue which is important for alpha-toxins. And we could recently demonstrate in a more complex model that indeed there are perilane quinones which are immunosuppressive, and we expect that they do the job maybe even in the bloodstream. So how can we say that? So we used a model on a trans well where we cultivated CACO2 together with HG29 cells, which form a mucus layer on top of that strong differentiated layer. And on the basolateral side, we placed the THP1 monocytes. Then the compounds were added to the apical side, and we detected here basolateral the impact on immune response. And we could demonstrate that that compound here, alpha-pyrilinol, which is an alpha-toxin without epoxide, really affects strongly the immune response on the basolateral side, and that represents the blood side. So that means we expect really effects here. So that was just published, that alpha-pyrilinol and also other alpha-toxins may be of importance here. So, and last but not least, I will quickly show you that these complex mixtures of alternaria toxins might also change your microbiome in the gut. So we generated a whole complex mixture of alternaria toxins and incubated different representative of different phylae. And what you can see here is the result in that scheme, that some of the cultures grew more, others were inhibited. So in total, we saw that we completely changed the composition of the microbiome. So they are expected to have an impact on your gut. So, and with that, let me summarize that a little bit. So alternaria toxins rarely occur as single contaminants. We see genotoxic, endocrine disruptive, and immunosuppressive effects. The genotoxicity strongly depends on the epoxide-bearing perilane quinones, and we don't know their fate at the moment, and we try to elucidate that. Since April 2022, there are indicative values in place for AOH, AME, tenoarsonic acid, and tentoxin, but these are only indicators for infestation, and we need more data on occurrence of the perilane quinones. So, and I have one last slide to demonstrate in human breast milk from Austria. What do we find here in the human breast milk? We have bovaricin, eniatins, we have Alternaria toxin, ochratoxin A, and cearalenone, all compounds I have introduced to you today. So this is still a topic. This is not Africa, this is Austria, a real Austrian breast milk sample. But don't think the Austrians shouldn't breastfeed. Also in infant formula, you find different types of mycotoxins we have investigated, and there you also have quite a mixture of toxins there. So is that a problem of plant-based foods? No. I showed you aflatoxins and ochratoxin A can go into dairy products and also in other products. And yeah, with that, nearly at the end, we need more mitigation strategies. We need awareness. We need to consider that we have a global food security issue here. And if we say, oh, we cannot import the contaminated stuff from Africa, then that means the kids there will eat it, and we have the uncontaminated ones. So we have to help globally to really raise awareness and to train people for mitigation strategies. So in the end, despite improvement, we still have an issue. Climate change is a big driver, will change everything. We have to consider these complex mixtures we take up every day, we might take up, and emerging mycotoxins are a topic which will keep us busy for the next decades. And with that, I'm at the end. Thank you.
Thank you very much, Doris. This was an excellent presentation, very clear, complete, and very comprehensive. So basically, we are surrounded by mycotoxin. But as I understand that we are living in sort of symbiosis with them, that we have to be aware of increasing their level by food transformation and of course global warming. That is very important to be aware of.
Yeah.
Because these mycotoxins are quite toxic. So I have some question already. So first question from Mohammed, there's two questions actually. Is there any, I'm reading the question. So I will do it for you. Is there any relationship between aflatoxin and lung cancer?
Not that I'm really aware of. That is not really clear cut. There might be conditions where you have a very severe wet building condition that you really inhale particles all the time with these aggressive compounds where there might be contribution. But the most important way is intake via food and indeed liver cancer.
Excuse me, can you shut down the microphone, please? Thank you. Then the second question for Mohammed is, how can we benefit from human biomonitoring and biomarker detection on human risk assessment?
So, well, there are many things you could mention here, and you really can calculate in a much better way on the exposure, and depending on what you are detecting, also conversion, and that's metabolic activation, metabolic inactivation. It depends on what material you analyze and what you really do analyze. So with respect to aflatoxin, there are several markers you could go for, and that gives you quite a good imagination how high is the level indeed of exposure. But you always have to consider the window, which would give you a respective insight into the exposure scenario.
Do you think there is an advantage of in certain population at risk to monitor the mycotoxin in the blood?
Yeah.
Is there an advantage for that to biomonitor this patient, or just to be sure if they are really at risk? Some populations that are highly exposed, no?
The population which are highly exposed, yeah, and you know, with our really genotoxic ones, I think you have to dissect between the ones which generate DNA adducts and the ones which have other effects. And we have this whole package of immune modulatory mycotoxins. And I think this is not well understood. So what that composition of compounds contributes to all these immune response to shots, yeah, so that this might be suppressed. So I think it would give us a more insight into the individual development of adverse effects and to correlate the real exposure to real effects. And we need more knowledge here.
So there is a question from Rosie. So does pulmonizing can be considered as a zoonotic disease?
Sorry, I didn't understand that very well.
Does pulmonizing can be considered as a zoonotic disease? Zoonotic, I mean, in animals. It is an epidemiology in animals or something. No, not epidemiology, an epidemic in animals. I think zoonotic disease.
Oh, I don't think so, but I'm not a specialist on zoonosis. But I don't think that it really grows in animals. But I have to admit, this is not my field. I'm not a veterinary medical person, so that's not mine.
I had a question from Anja. Where is the question from Anja? Okay. So thank you for your great talk. I wonder if there is a reliable mycotoxin detection assay for consumers to test food at home. It's an interesting question. Have a rapid assay at home. That's interesting.
Well, they're very expensive quick tests, which are in use in certain parts of the world to help farmers or producers, but this is not very reliable. You have to consider that maybe you got a glimpse how very, very different these different structures are. And there's not one test at the moment where you really can have this diversity and see that in a quick test. That's just not possible.
But do you think that one day for some of the mycotoxin, the consumers will ask for it? To test it? Or to be tested?
Yeah, maybe. Maybe. That's I cannot exclude it, yeah. But that will be, it's difficult, you know. Consider you have these oat flakes for breakfast, yeah. This is a dry material. How do you test it? Because you have to water it because you have to make it available and in the right material. So that's kind of difficult.
But right now, you don't have any kind of legal obligation to write on the package the amount of mycotoxin.
No.
That's a legal requirement.
That would terribly alarm all consumers. This is containing mycotoxin.
One day we'll ask for it. One day they will.
No, but of course you have to keep the legal limits for all the regulated ones. Yeah.
So I have a question from Luc. So Luc is asking a question about the Aspergillus-like micromycetes and articular disease, rheumatoid arthritis. Do you know if mycotoxin are involved? It's Aspergillus-like micromycete. You see, it's a fungi. You don't know about that?
No,
No.
not that I'm aware of, no.
So we go to the next question from Manel. Does zearalenone, with its endocrine disturb effect, can perturb neurocomportamental children? No.
Could you repeat, Mark?
Yes. I'm sorry, it's my accent. Does zearalenone, okay,
Yeah,
with its endocrine disturbing effect,
yeah.
can perturb the neuro of the development of the in the children, neurocomportamental development.
Well, it's endocrine disruptive. So you the question is in which time window and how high is the exposure of estrogenic compounds the fetus is exposed to. And it may indeed occur that that starts quite early. We know from pigs that fertility is impaired with a high consumption of zearalenone, yeah? And it's well known that exposure to estrogens in the early development of children might really induce changes to early puberty, for example, or changes in the male development. So there are impacts, yeah? And with that background, we do have the regulation which are in place at the moment, yeah, where these compounds are really regulated and controlled, and that's very important.
Okay. I have a question from Emanuela. So, can pesticides be of help in reducing fungi or by reducing food stress, resulting in a reduction of mycotoxin?
Yeah, that's a very good question. We are discussing that since quite a while. So the question was, is there a choice between we have the biofood with mycotoxins, without pesticides, and otherwise? But that's not always the case, actually. So sometimes that might occur that if you have enough pesticides, the fungus doesn't grow. But when I can cite the actual case we do have here in Austria from last month, where you had T2 and HT2 on oat flakes, yeah? Then the only samples which were free of T2 and HT2 were the bio ones, yeah? And then we had even some of the samples had T2, HT2, and pesticides. So this is not a general rule that the use of pesticides would indeed prevent from fungal growth.
Okay, because she had a complementary question that have difference in biological products versus regular products that are grown with pesticide be reported in the level of mycotoxin. So there's no strict report showing that with pesticide you have less fungi, less mycotoxin, and without pesticide. Yeah.
From my point of view, you cannot have that as a general rule, no.
Okay.
And you have to consider that some of the pesticides do have also estrogenic impact, no? And then if you indeed still have fungal growth, then you might have combinatory effects.
Then a question from Tanya. How good is the monitoring of foods for mycotoxin? It all sounds very worrying.
Well, you cannot control everything. I think there are quite good plans in place. Different countries are different in their strategies to control the regulations. I think the regulations we do have at the moment in the EU are okay. And I think the countries do their best to really stick to it. We have to be aware that we have these global food production chains and these streams of food coming from wherever. And that changes a lot, and to the good and the bad. And I know that in Germany, one of the biggest labs for food control is at the airport in Frankfurt. Yeah, because all that stuff comes in. And the same is also true for the Netherlands, for the big harbors, for example, yeah, that the overseas cultures need to be controlled. So you can't control everything. And I think the consumer has also to have some awareness. So if you buy figs and they have this white thing on top, then you have to be critical, yeah,
Yeah.
of course.
We need to, I mean, the people need to be more educated regarding this.
Yeah,
People
yeah, I
need
think so. I think so. I have to admit, I don't eat canned tomato products anymore since I do these analysis. So but this is something. Well,
Okay. So I've got one last question. So it's rare to find single mycotoxin in food. Is there any studies about the mycotoxin mixture effect?
there are a lot of these studies. So, so one of the first groups working on that were the group of Isabel Oswald at Toxalim, and I think meanwhile a lot of people have contributed here. Also, we have done a lot of studies on that. Of course, most of these studies are in vitro. There are some synergistic effects, but most of the effects in the end, when you look for the biological relevance, are more additive. There are some effects which are more potent when it comes to hormonal cascades, yeah, and some when the combination of compounds trigger over a certain rim, you might have an overwhelming effect, of course. I think this is not only a mycotoxin problem, this is an exposome problem. So we have to consider the whole exposure. And that means when you start in the morning with your coffee, with your oats, with your rye bread, and you go on over the day, you have so many different compounds. And this is not only mycotoxin, this is also pesticides, this might be PFAS, this is a lot of things, and they work together, and we have to consider also here the mixture effect.
So thank you very much, Doris. Now it's past it's 2:02. So thank you very much for this excellent presentation. It was really timely to have this webinar on food toxicology, because I think people are not enough aware of food toxicology. I think that's very important, and the mycotoxin is really an issue. So I did publish one paper on the effect of deoxynivalenol done on the apoptosis of telanophytes long time ago. So I know it's a big issue. So thank you again, and thank you for the people asking questions, and thank you for the people who attend the meeting. And so see you soon. See you next Friday, Doris. Huh?
Yeah. Okay. Thank you so
Bye-bye.
much.
Have a nice day. Bye-bye.
Bye.